Hi guys, welcome to Fast and by Fungi podcast. We're back for episode 12, where I'm going to talk about fungal bioremediation or mycoremediation, which is the idea that fungi can help clean up toxins, plastic, radiation, all sorts of stuff in our environment. But I'm going to approach this subject from a very sort of scientific standpoint, because I think there's a lot of hype that gets thrown around the subject. And occasionally the science can't quite keep up with what's actually happening in nature. And a lot of what we're looking at is just trying to look at the interface of fungi and nature and what's happening. And scientists are sort of mining natural sources to figure out what's going on and to find organisms that could potentially help detoxify things. But journalists see these headlines and say, oh, we've solved plastic waste, we've solved problems with oils because somebody somewhere has figured out how to do one thing in the lab. And I want to take this podcast episode to kind of approach the idea of bioremediation and positives, but also some of the things that have maybe been overhyped or overpromised when we think about bioremediation. And I'm going to start by just quickly a couple housekeeping things here about the podcast. I am back on the third week with new audio equipment. I've got a canister mic for YouTube, so hopefully it's working this time and you guys can hear me. I've got a little clip-on mic because I was kind of upset with the way that the headphones were working because the sound seemed to be going up and down. So I appreciate you guys sticking with me and listening to the podcast, even though the audio hasn't been totally perfect. So the other thing that's exciting is Colin, the app that I'm broadcasting this podcast on, has recently added syndication RSS feeds for Spotify and for Apple. So hopefully soon you'll be able to watch this or listen to this on those platforms as well as YouTube, as well as here on Colin. So I'm excited to talk to you guys here about bioremediation. It's a really exciting idea that we can use nature to help clean up the damage that we've done to nature, basically. And I think one of the things that people forget is that we have a lot of bioremediation that happens somewhat naturally in our environment. Wetlands are a great bioremediator. That is where a lot of the detoxification in nature happens. If you think about Earth as sort of like an entire human body, the wetlands are often like the sort of the liver or the kidney that's doing a lot of the filtration and the detoxification of things that happen upstream as it comes into the ocean. And sometimes the most effective way to bioremediate stuff is essentially to let nature take back over the areas that we've done damage to. And if we have destroyed a site by doing mining or having a nuclear explosion, whatever it is, letting nature get back in there and kind of just do its thing is often the most effective way to end up bioremediating it. But that becomes a problem if we have stuff like oil spills, where we've just spilled so many contaminants into an environment that there's just no way for the life and nature to kind of keep up, at least not on a time scale where we'd like to see like clean waters again within our lifetime, instead of having oil floating around forever in the ocean. And at this point, it's probably too late with some of the oil spills that have happened. But we can talk about the potential for fungi as bioremediators. And I think the thing to keep in mind with that is fungi are absolutely everywhere. I'm not just talking about mushrooms, but fungi, so molds, yeasts, filamentous fungi. There are things that are on every surface all over the plant. I have probably mold spores here on my table in my home. There's also, yeast and mold spores down in Antarctica, in Arctic environments, in tropical environments, in desert environments. Fungi are absolutely everywhere, and they capture so many different environments. And because of that, that means there's great promise for them as bioremediators, because they are at every interface in life. And so many of the places we look at to say, man, we need to clean this up, there's already bacteria and fungi that are there that are surviving to some degree. And that's a place that scientists have been going and looking for organisms that could help us bioremediate some of the damage that we've done. So again, I'm getting comments here on YouTube from people saying the audio is not great. Sorry, deal with it as best I can do. Trying to stay focused on the podcast, and it is what it is. This is what I get for doing a live stream. Sometimes things don't work out perfectly, and that's just what it is. So let's talk about positives as fungi for bioremediators and the things that we know they do well and why scientists are so excited about using them. And a little bit later, I'm going to get into sort of like a skeptical review of why I think some of the work that's been done with bioremediation has been a little overhyped or a little overpromised. And we'll talk about some actual concrete examples that have been done and worked on. And we'll talk about some of the approaches that scientists are taking to do this kind of work and what it might take to actually translate what happens in the lab into like real world, real life environments and see how that works. So fungi, like I said, they're ubiquitous, they're everywhere, which is great. That helps a lot with figuring out, would this organism grow here? Like, yeah, of course it does. It grows here. So we just have to find the organism that is in this environment already and then test its ability to potentially deal with the different types of stresses that's experiencing because of the toxins or the pollution that we put out there. And a lot of the pollution that I'm going to be referencing are like different types of plastic polymers. There's radiation. There's a lot of like pesticides and herbicides and hydrocarbons in pharmaceuticals, endocrine disruptors, all this huge class of these sort of persistent trace organic contaminants that are just all over our planet and are wreaking damage on living organisms, including humans. And this is part of why we've seen this massive jump in cancers and autism and other things like that, because we have so many of these sort of trace organic contaminants, and we don't fully understand the biological activity of these things. But fungi are really good at kind of seeing these things in their environment, understanding that that compound is probably toxic to them, and they're dealing in real time at like a microscopic level with some of these contaminants in a way that like our human bodies cannot, because fungi are able to change their gene expression much more dynamically. And because they're faced with these toxins kind of like face on, they're having to learn how to like evolve and deal with them or adapt to them in ways that like our human bodies are just not equipped to do. And so that's why turning to fungi to understand this bioremediation has been sort of so exciting from a scientific perspective. But fungi are really good at growing into and invading substrates. And so they can grow in soil, many different soil types. They can get down into areas where there is like seepage of toxic chemicals and stuff like that in a way that like trying to excavate or do some other process just wouldn't be feasible. And so fungi can spread across the environment, like I said, be ubiquitous. They can connect source and sink, i.e. they can facilitate the transport of nutrients and sugar across like mycorrhizal networks. And so they can also transport organic contaminants across, networks as well, not just nutrients. They, in forming those mycelial networks and microbial associations, can help facilitate the movement of bacteria. And that's really important too for microremediation, because I think one of the things we're looking at with fungi is, well, fungi have the ability to digest or detoxify particular elements that might be toxic. They don't necessarily carry the entire arc of something from being toxic to being non-toxic. They're just taking part in one little step of this detoxification process. And as part of what's called ecological succession, bacteria will also play an important role in detoxifying many of these compounds that are these trace organic compounds, contaminants. And so it's going to take, as you would say, it takes a village. It takes a whole microbial succession of different organisms, fungi and bacteria, to detoxify and sequester some of these organic contaminants. So again, having the ability to transport bacteria around and have a rich, diverse microbial community fostered by fungi is really important when we look at this type of remediation that we're hoping to do with these biological agents. So the main way that fungi are attacking and dealing with the stress of these trace organic contaminants is through their extremely powerful cell enzymes. So one of the things that's been looked at specifically with regards to like digesting oil spills or hydrocarbons or plastics is the enzymatic capacity of white rot fungi. So those are primary saprobes, stuff like oyster mushrooms, Ganoderma, lion's mane, things that are able to degrade lignin. So lignin is this fairly complex, multi-ringed structure that's in wood that cross-links. Cellulose is lots of parallel fibers. Lignin is cross-linking all those cellulose fibers and giving wood its tensile strength, making wood very strong. So white rot fungi have this amazing suite of enzymes that they've evolved over millions of years to digest lignin. And like I said, lignin is these very complex polycyclic rings that don't look totally unlike different types of hydrocarbons, especially these like polycyclic aromatic hydrocarbons that are very toxic and very hard to break down. So these white rot fungi can adjust some of their enzymes, stuff like oxyreductases, and these enzymes often have sort of low specificity because they're really just trying to deal with like any organ or any like molecule that looks kind of like lignin, which is useful for us because we want them to degrade a spectrum of different things rather than just like one very specific thing. And so that low specificity can actually be like a helpful thing as we look at fungi as our allies and helping to deal with bioremediation of toxic stuff. But beyond the enzymes, there's also the ability to sequester or sort of reduce a lot of these toxic compounds. And so what they're doing is they're taking them up, and as part of their biology, they're making them a little bit less toxic, or they're sequestering them in one particular part of the fungi to make them not toxic to the rest of the organism. And actually what usually ends up happening is a lot of the toxins that get absorbed by mycelium end up getting pushed out into the mushroom. And this is a problem from like a foraging standpoint because there's certain mushrooms that are prone to accumulating heavy metals. Mushrooms are pretty good in their mycelium with absorbing heavy metals from soils, and these heavy metals are still toxic to the mycelium, so they'll end up maybe tacking on something to the heavy metal to make it slightly less toxic, but they'll push all those heavy metals up into their fruiting bodies, their mushrooms, to the surface of the soil. So it's not in the body of the mushroom down in the soil, but instead it's above ground. And then you have this concentrated little thing of bioaccumulated heavy metal from all the soil below. And as humans, if we're foraging mushrooms, we end up with a tasty looking mushroom. We don't really think like, oh, this might be full of lead because this mushroom grew on the side of a road where 50 years ago somebody was driving cars with leaded gasoline, and all this runoff has brought the lead into the soil. But that can happen. And so there's certain species, like I think Caprinus comatus is one that grows really commonly in like urban areas, but it's also known for accumulating heavy metals. And so kind of a shame because I do love Caprinus comatus, but I don't gather it on roadways because you can have lots of lead and heavy metals in your mushroom, and you wouldn't really know. So that's kind of a scary thing to think about. But again, it's probably better overall to have the mushrooms there, the mycelium doing its thing, as you're still sequestering and helping to slowly detoxify some of these heavy metals and these like trace organic contaminants. So some of the fungal enzymes that are there, and this is stuff that's not just white rot fungi. This is a lot of fungi have the capacity to do this. We have Aspergillus, Fusarium. There's all sorts of filamentous fungi that have been looked at. Mucor molds and even Glomeromycota, the tiny little things that live on the roots, create mycorrhizae, but they don't actually create mushrooms. These have all been shown to be really, really effective at sequestering trace organic contaminants and at detoxifying things as part of an ecological succession. They're taking only one step in the decontamination pathway that needs to happen to detoxify these things, but they're still really important in that that process, that arc of what's going to happen in the soil naturally. So some of the fungal enzymes that we're talking about are like catalases, laccases, peroxidases, lipases, lignases. These are all things that can help break down different types of organic molecules. Specifically, what they're looking at is like breaking down carbon linkages and very specific types of carbon linkages that are in the food that mushrooms would eat. They're in wood, they're in different types of substrates. But the important thing to keep in mind is that mushrooms, particularly, let's talk about oyster mushrooms, a good thing here because they've been used a lot for bioremediation, is that if we put a mushroom in a situation where it has limited food and we give it a weird carbon substrate, it's probably going to figure out how to eat that weird carbon substrate because that's what fungi are really good at doing. They are opportunists. They are always looking for a way to get in there and figure out how to make life happen. And if a door opens for them, they're probably going to go through it and explore it, because that's kind of the nature of mycelium. It's always like creeping and exploring new avenues, even metabolically, to say, hey, I just had this whole new carbon source open up to me. This wood's getting a little stale. Maybe I'll try this thing. But they don't naturally produce these very diverse sets of enzymes unless they have to, unless there's a reason for them to kind of be more exploratory. If they have lots of wood and they're pretty happy, they're probably just going to eat that. But if you give them hydrocarbons, you can force them into eating hydrocarbons if you leave them sitting in a lab long enough with hydrocarbons as really their only like food source, or oil or things like that. But the mushrooms don't inherently like that. And a lot of fungi are similar. They don't inherently like the weird things they end up eating. They're eating them because they're in environments where they've been exposed to toxins, different plastics, radiation, stuff like this, and they're having to figure out how to make life work. So it's not like they're preferentially seeking out these weird situations and these substrates where things are toxic. They are just adapting opportunistically to new situations. And so part of the reason we've been isolating so many different fungi that have the capacity to break down these trace organic contaminants and plastics and radiation is because we have put so much pollution out into the world that there's really nowhere on this planet that doesn't have some trace level of the pollution that we put out there, whether it was like the nuclear fallout from testing nukes in the 50s, whether it was the fact that like plastics and microplastics are pretty much everywhere in the world and inside of every single organism at this point. The fungi are just dealing with it as it comes. You know, they aren't like going out of their way to eat these things that are weird. They would prefer to keep eating what they evolved to eat originally. But as our environment changes as a function of human activity, we are seeing more and more fungi kind of get into this weird niche where they're like, okay, well, I guess I'll take a little bit of DNA damage because that's the pollution is coming at me, and I'm going to try to retool my fungi enzymes and my metabolic pathways to deal with this new type of stress and try to detoxify it to some degree so that I can continue living where I am, because fungi aren't generally modal. They can't swim around, so they can't move away from the things that might be hurting them. So they just have to change their own programming to help them kind of adjust and adapt in this very dynamic way. So there has been work done. Paul Stamets is a guy super famous for doing some experiments where he was able to show that Pleurotus oyster mushroom mycelium could digest, hydrocarbons in a lab, or cigarette butts in a lab. And he proposed all these ideas of like, oh, well, we'll take this phenomenon, we'll put all these oyster mushrooms out in blocks and clean up oil spills, and we'll clean up wastewater and all these other things. And I think if you look up, fungal bioremediation, you find a lot of articles from like the late 2008 through like 2014 kind of thing, where everyone's saying, oh, mushrooms will save the world. They'll save the world. We've solved it. We've solved it. And you don't find that many more recent articles. And I think that's because Paul Stamets kind of came out and did this work like 10, 15 years ago. And everyone's like, oh, great, we've solved oil spills. The problem is what he did was a very specific lab situation where he took, sort of an idealized mushroom or mycelium, gave it a difficult substrate, and gave it a long time to adjust. And then was able to show that he could make this mycelium adapt to eating a weird substrate. But if you take that same mycelium and throw it into nature, it's going to have other food sources available. And so it's not going to be able to do much. And that's a really important thing, because when you think about fungi's ability to deal with stuff, it's really important that we consider the environmental conditions, like the pH, the temperature, the humidity, the soil type, the water availability. There's so many factors that go into whether or not a mushroom or fungi will be able to do what you want it to do in a natural situation. And most of the work that's done, and if again, you can Google bioremediation, I found, 23 papers of really interesting, cool results. I was like, oh, cool. Mushrooms can eat polyurethane and polyethylene, and there's a fungi that can digest plastic and water under aerobic or anaerobic conditions. They can do different things. all very, very, very cool stuff. And there's some real promise and potential out there. But the problem is almost all of those things are, hey, in a bioreactor, I gave mushrooms this idealized situation and I gave them a single contaminant at a time. And I was able to say, hey, if I limited all of the carbon sources, would the fungi grow on this weird thing I gave it? And the answer was yeah, because fungi are really good at doing that. They're really good at adapting and being opportunistic. But the problem is, if you put them in a more natural situation with a broad variety of these different types of contaminants, there's rarely do we ever have just a single contaminant. It's usually lots of different contaminants in complex mixtures. And you're putting them in an environment where like the pH, soil type, and temperature and stuff aren't as well regulated. The fungi aren't necessarily going to respond the way that they did in a lab. They're going to do what is best for them in that moment, which is to survive and create offspring and keep growing in that environment, which may not mean digesting the hydrocarbons or the plastics or whatever it is that we want them to digest. So in a lab, we've shown all sorts of really cool activity, but in real situations, it's much, much more difficult to get mycoremediation, bioremediation happen in the ways that we've sort of idealized it and in the ways that have been like proposed and promised to us by some of these famous mushroom people. And it's not to say that we shouldn't keep trying. We absolutely should. But kind of like I mentioned a little bit at the beginning of the podcast, like sometimes the best thing to do isn't necessarily to, take one organism and try to put it out in blocks to solve an oil spill. It's like we should really just let biodiversity take back over and let nature have at some of these things, because it's already evolved in many ways to deal with different types of stresses. And at a microscopic level, there's a whole bunch of fungi and bacteria that are starting to kind of adapt to these weird things that we put into the environment. It's not necessarily a good thing. We shouldn't have put so many microplastics, endocrine disruptors, pesticides, herbicides into our environment. But evolution on a microscopic level happens much faster. We have single-celled microbes, and their generation time is like 4 to 6 hours instead of it being, let's say humans are 30, 40 years generation time. So they're able to adapt much, much faster and more dynamically. And we're seeing that when we go to areas and we're isolating organisms and we're looking at their ability to degrade things. So let's see, I looked up a couple of real life examples here, things that I thought were pretty cool. So there was a Nigerian oil fields where they went and isolated some aspergillus and some mucor molds. And they were able to see that they were able to break down some hydrocarbons, again, in a lab situation, but these were things that were pre-adapted to living in like an oil field. And the problem was they only partially broke those things down. And this is kind of what I'm getting at, is there's, you'll see these headlines that say, oh, there's a marine fungi that can digest plastic. Cool. So I looked it up. Zalarian meritonium, something like that. And it can degrade polyethylene. Cool. Sounds great. Problem is it degrades it into like smaller units of plastic. So there's a fungi out there that's digesting plastics in our ocean, but it's just making more microplastics. And is that really a good thing, right? Like plastics will break down eventually on their own into microplastics with enough like heat, time, and UV, but there's a fungi out there that's accelerating that process. And so maybe it's a good thing, because we could take, let's take this fungi, put it into like a big bioreactor with a bunch of this polyethylene stuff, and we'll create smaller monomers. Maybe we can use those for something. But in like a natural situation, this fungi is growing on like floating plastic in the ocean and just creating more microplastics, which are getting deeper into like the food chain, the fish, the sponges, the inverts, everything is suffering because we have all these like weird little units of plastics floating around that can have damaging biological activity, because these small units of plastic can act as free radicals. They get inside of cells, they get inside of living organisms, and they are causing oxidative damage to the DNA of things. And that's part of why we're seeing not seeing like elevated rates of mutation. We're seeing elevated rates of cancer, because unfortunately, I think, X-Men has, my generation grew up on X-Men. I thought it'd be really cool. It's like, cool, I get like a dose of radiation, I'll turn into a mutant, or I'll get like spider powers. Like, no, you get a dose of radiation, you just get cancer. There's no cool, mutations that happen that would somehow make you superhuman. You're just going to get cells that have forgotten what the cell cycle is, and they're going to grow out of control. And that's basically what a tumor is. So I think there's some incredible stuff that's happening. There's another one. I'm going to struggle to pronounce these names, but bear with me. It's Pestaloteopesis microspora. So this was an endophytic fungi isolated from Ecuador. And the group showed that it could digest polyurethane in anaerobic environments, which is super cool. And I think that's one of the ways we could actually look at using fungi to effectively tackle problems with plastics, would be to get some strains that work really well under anaerobic conditions. That means no oxygen. You put them into like a big digester with maybe like a soup of plastic, and hope that this fungi is able to bioconvert enough of this plastic into a different form, or less toxic form, or maybe a reusable form. And I know that there's already, I think, bacteria and some other things that are being using anaerobic digesters to digest different forms of hydrocarbons. And you can, turn unusable plastic into fuels using bacteria in an anaerobic digester. And so that's very different when we think about, it's bioremediation, but we're transforming one product into another product with an organism, which is not what we'd think of traditionally as bioremediation. That's more like production. But we could use the lens of producing value-added products from waste as a way to encourage people to actually do more recycling, do more effective recycling, because a lot of the recycling we currently do is not sustainable and isn't bio-based, and is really just about like making us feel better, the fact that we're constantly polluting our environment with more and more plastics and more hydrocarbons, and not actually doing anything effective to change what's really happening. There was some work I looked at with spent mushroom substrates, I think from like Pleurotus oyster mushroom type things. So this is like, spent mushroom blocks that have been used in mushroom farms. And then they were like running wastewater through this, and they saw a reduction in phthalates in the wastewater from being run through like mushroom mycelium. And so that's the question of like, is it really degrading the phthalates, or is it just like binding and absorbing them and sequestering them? Either of which is okay, but it's also not something that's like necessarily scalable. And again, this was like done in a lab setting in a bioreactor, and they're like, well, is this scalable to like municipal waste systems? They don't know. There was a great article I was looking at that was talking about the removal of selenium, which can be a fairly toxic heavy metal, from wastewater, and it was looking at like municipal wastewater versus like industrial wastewater. So one was sort of like more tame versus the really industrial, which is pretty dirty. And they essentially saw that the municipal wastewater, the fungi were actually pretty good at removing the selenium from that. But in the industrial wastewater, which was far more complex and a far more rich media, lots more stuff in there for the fungi to eat, the fungi weren't detoxifying the selenium because there was so much more carbon and other nutrients available. And that really gets at what I was talking about with like the Paul Stamets oyster mushroom thing, is that he was like, yeah, look, we can clean up the environment with oyster mushrooms. And like, yeah, but if we put oyster mushrooms into a natural environment, they're going to go ahead and want to eat wood and other things. They're not going to want to eat these weird carbon substrates. They're not going to want to eat petroleum or cigarette butts in a natural environment. And there's going to be other things available for them to eat. And, I think it's an important thing to remind ourselves, is that it's really easy to say, look, we got this result in a lab, but we're not going to see it in nature. And so how do we take what we've learned about the capacity of these organisms to do detoxification and translate that into an actual like ecological strategy, where maybe it's not just a single organism, but it's like a suite of different organisms that are creating that arc of the ecological succession that can specifically degrade high concentrations of particular trace organic contaminants that are in environments. So again, much like letting a wetland or a forest ecosystem kind of take back over areas that were polluted, you'll see a reduction in a lot of these trace organic compounds if you just let life do its thing. We can help it along by understanding specifically what organisms might be good at doing the succession and seeding the environment with high populations, high bioloads of these organisms that also have been pre-adapted. So we can take organisms, pre-adapt them in a lab, or isolate them from an environment where they've pre-adapted themselves genetically through a series of maybe mutations or genetic changes in cell cycling and cell regulation, and then apply them to different situations where maybe the organisms in that niche haven't yet adapted to that. So we can say, hey, we took this bacteria and this fungi that evolved to eat radiation in one spot, and we took them somewhere else and put them in at a high load, and now they're helping to detoxify the radiation here, even though nothing here had yet evolved to deal with it. And so that's where I see the promise of this stuff. But there's just so far to go in terms of understanding the science. And I'm not trying to be critical of bioremediation, but I do think sometimes it's overhyped. And again, if you read the headlines, what people have written, it makes you feel like we've solved the problem, which is so far from the truth. We are so deep in the weeds with not really understanding what the heck is going on biologically. Even, if you go and look, there's all this press about Cryptococcus neoformans, which is that was the fungi that they isolated from Chernobyl that can somehow grow in radiation. And it's not like it grows really well. It's a very sick organism. But what it's done is it has harnessed the ability to generate energy from radiation. But radiation is inherently very damaging. And so they did this big meta-genome analysis and transcriptome analysis of the Cryptococcus genome. How is this thing surviving? And essentially what they saw, and they don't totally understand it, but they saw massive amounts of changes in the DNA repair regulatory network. And so they were looking at cell cycling proteins. They were looking at enzymes that go around and fix DNA. When you have DNA that gets damaged, you'll get perturbations in the double helix. And so there's enzymes that scan that double helix looking for little structural perturbations. And then they'll go ahead and resect it and fix it and just kind of continually curating the genome to make sure it's robust, and that's what Cryptococcus has gotten really good at doing, is massively upregulating the number of DNA repair enzymes that are active within its cells, and so that's how it's resisting so much DNA damage. Otherwise, it would just crap out. And I think there's some degree in that's really cool, but it's not like this organism has survived, evolved to some that's a thing you see a lot in nature, is things evolve to be just good enough to live. And it's not like they're optimized, right? And we as human beings could think about trying to genetically engineer some of these organisms to optimize them to deal with plastics, to deal with radiation, to deal with hydrocarbons. But if we do that, there might be unforeseen consequences, because you start messing around the genomes of things, and those genes could get into other organisms. And say, here's a test case just to think about. Say we took one of these fungi that seems to be able to degrade different types of plastics, and we said, okay, we really want this to get out there and digest lots of plastics. So we're going to go ahead and tool around with its genome. We're going to adjust the genes. We're going to change the regulatory networks. We're going to genetically engineer this fungi in a facility to break down plastics and help with recycling. We use a couple different fungi and bacteria as part of biological succession to create this more effectively bio-recycling plastics. And this, to me, is sort of maybe the best case scenario of how we could be using this. But let's say somebody gets a little careless one day, and they take a little bit of this fungi material out of the facility, and it gets into nature. Now we have a fungi that can eat plastic in nature. Sounds good, right? We're going to start degrading plastics in landfills and all over the place. But there's so much plastic that maybe that fungi gets out of control, grows everywhere, across the planet, and then it starts attacking our plastics in our phones, plastics in our electronic, plastics in our hospitals or medical equipment. And suddenly plastics are rendered useless, obsolete, because there's a fungi that there could be negative consequences potentially to actually evolving a fungi that could on its own fully break down plastics. Or, what is also happening is just there's fungi that break down plastics, but they're breaking down plastics into smaller plastics. And so they're only detoxifying like one step of a whole process, and helpful because we could still be, sequestering and digesting certain toxic compounds, but then leaving unintended consequences of producing other toxic compounds or spreading those around in our environment in sort of unforeseen ways. Um, and so I'm a little skeptical of bioremediation simply because when I hear people talk about it, they talk about it as if it is just this like glorious future where all problems will be solved by biology. And I love that idea. I really wish it was true. I'm waiting to see how the science works out a little bit and hope fungi and bacteria to work as bioremediators. But I think also in calling them bioremediators, we're minimizing the role that they've had for billions of years on this planet as terraformers, as agents of change and ecological engineers that have created the world as we see it and as we kind of care about it. And what needs to be done less than like potentially isolating all these things can, eat radiation, is to just give nature a chance to survive. There's so many areas where humans have done tons of damage. And if we could go back in and just reseed, let nature take back over. That was kind of what happened in Chernobyl. They just like generating energy off of breaking down plastic into even smaller units of plastics. And then what happens to those? It's a complex that are going to come as sort of reckonings, and we, at the moment, we can't really possibly imagine. They seem sci-fi. They seem unreal. But there will be points we hit, metrics we hit within like the human milestones of where we go over the next 50 to 200 years. They're looking at really severe effects of climate change, crazy unstable weather, oceans rising, and we're going to have to look at bioremediation as a core way that we deal with rehabilitating environments. And it's not just bioremediation by like fungi and bacteria to get rid of some of these trace organic contaminants. It's replanting mangroves to save coastal areas. It's rehabilitating wetlands and the huge diversity of bacteria and other things in there to help filter out water that is toxic, that is coming off of our land from all of the um, pesticides and herbicides. We're going to have to stop using so many pesticides and herbicides and look at using, you know, biological. But you know, there's this healthy ecosystem that has grown back up around Chernobyl because we just left it alone. And you know, I think there's real some lesson to be learned there, that it's not going to just be like one super fungi that cleans up the world for us. It's not going to be just one thing that we've engineered or we've done. We need to rely on nature and, and the capacity it has to be adaptive, to be opportunistic, to deal with some of the damage we've done. We also need to start looking at our own consumption habits and reduce the amount of waste that we put into our environment, because at some point it might get overwhelming. And